Bismillahirrahmanirrahim. Assalamu alaikum. I welcome all of you for today's lecture of physiology of the thyroid gland. I would like to appreciate your interest and dedication for the learning initiative of mmthub.com. Thyroid gland consists of secretory part called the follicles, which are filled with the hormone storing thyroid, and the parafollicular C cells, which secrete the calcitonin. Synthesis of the thyroxine. Thyroid hormone synthesis starts from the step of iodide trapid by the thyroid follicular cell via the sodium iodide importer. This iodide is diffused to the apex of the cells and then carried to the thyroid. In the thyroid, there will be the process of oxidation of inorganic iodide to iodine in the presence of thyroid peroxidase. On the other side, thyroglobulin is formed in the thyroid follicular cell and then released into the thyroid follicle by the process of exocytosis. There is iodination of the thyroglobulin with the iodine and lead to the formation of the diiodothyronine or monoiodothyronine in the thyroglobulin molecule. If two diiodothyronine molecules combine, it will form the tetraiodothyronine T4, and if one monoiodothyronine combines with diiodothyronine, it will form the triiodothyronine T3. This thyroglobulin is then carried from the colloid into the follicular cell by the process of endocytosis in the thyroid follicular cell. This thyroglobulin then fuses with the lysosomes and process of proteolysis occurs, which causes the release of the T4 and T3 into the circulation, while the diiodothyronine and monoiodothyronine are released in the thyroid follicular cells. This diiodothyronine and monoiodothyronine then undergoes the process of deiodination to yield tyrosine. With this, tyrosine is again incorporated for the formation of the thyroglobulin. As far as T3 is concerned, T3 is also formed from the mono-deiodination of T4 in the thyroid as well as in the peripheral tissue. This is the slide which is summarizing the steps of the thyroid hormone synthesis as we already discussed. Secretion of the calcitonin. Calcitonin is secreted by the extrafollicular or parafollicular cells of the thyroid, and it causes the lowering of the blood levels of calcium and phosphate ions when they are too high in the circulation by the process of increasing the rate at which the calcium is stored in the bones and excreted in the urine. Also, calcitonin secretion is regulated by the negative feedback involving the blood concentration of the calcium. Regulation of the thyroid hormone. Thyroid hormone regulation is dependent on the thyrotropin releasing hormone from the hypothalamus, which stimulate the release of the thyroid stimulating hormone from the anterior pituitary gland. This thyroid stimulating hormone then acts on the thyroid gland to cause the release of the thyroxine. Thyroxine has an inhibitory negative feedback effect on the thyrotropin releasing hormone and thyroid stimulating hormone. In this regard, T3 is more potent than the T4. Cold increases and heat decreases the thyrotropin releasing hormone secretion, while the stress and the glucocorticoids inhibit the thyrotropin releasing hormone secretion. This diagram is also summarizing the regulation of the thyroid secretion and the role of the hypothalamus anterior pituitary in the regulation of the thyroid hormone secretion from the thyroid gland. When the homeostasis is restored, the increased T3 and T4 concentration in the blood has an inhibitory effect on the thyrotropin releasing hormone and then on the thyroid stimulating hormone to cause the decrease in the production of the thyroid hormone from the thyroid gland. Action of the thyroid stimulating hormone. Thyroid stimulating hormone increases the iodine uptake by the gland, thyroglobulin formation, T3 and T4 formation, endocytosis and release of the hormone, hypertrophy and development of the gland. It also causes the cyclical change of the gland size in the females. All these actions of the thyroid stimulating hormone cause the increased production of the thyroid hormone from the thyroid gland. Actions of the thyroid hormones. Thyroid hormones are lipid soluble and pass through the cell membrane easily. T3 binds to specific neutral receptor protein with an affinity that is tenfold greater than the affinity of the T4. Thyroid hormones stimulate the oxygen consumption in almost all cells of the human body. Thyroid hormone has a stimulatory effect on the rate of hepatic glucose output and peripheral glucose utilization, hepatic metabolism of the fatty acid, cholesterol and triglyceride. It has also stimulatory effect on the synthesis of the important proteins, sodium potassium ATPase pump, respiratory enzymes, erythropoietin, beta adrenergic receptors, sex hormones and the growth factors. It also causes the stimulation of absorption of carbohydrates in the intestine and gut excretion of the cholesterol. It has a modulatory effect on the female reproductive function. The thyroid hormone and the catecholamines work together in metabolic acceleration. Thyroid hormone increases the cardiac rate and cardiac output as well as the respiratory rate. It causes the high basal metabolic rate and peripheral vasodilatation. This vasodilatation forces the cardiac output to increase, and if the cardiac output is insufficient to match the vasodilation caused by the thyroid hormone, it can lead to a condition of called high output cardiac failure. Next is the action of the calcitonin. Calcitonin inhibits the bone resorption by blocking the parathyroid hormone receptors on the osteoclast. This result is an extremely effective lowering of the plasma calcium and phosphate levels. Calcitonin is important in bone remodeling and in treatment of the osteoporosis. Administration of the calcitonin leads to a rapid fall in the plasma calcium levels. Calcitonin is a physiologic antagonist to the parathyroid hormone and it inhibits the calcium liberation from the bone. But calcitonin reduces the plasma phosphate levels just as the parathyroid hormone. Thank you very much for the today's lecture. In the next third lecture we will discuss the pathology of the thyroid gland.